Good day, future doctors. This is the first PowerPoint presentation on the series of lectures on cestodes. Both cestodes and trematodes are classified under the phylum Platyhelminthes. It's because their bodies are dorso-ventrally flattened. What makes the bodies of cestodes different from that of trematodes is that the bodies of cestodes are segmented. This is a picture of a tapeworm. Take a look at its body. It's dorso-ventrally flattened. And you can also appreciate the presence of segmentations, which are absent in the trematode group. This is another picture of a cestode. You have here a dorso-ventrally flattened, segmented body. And since the bodies of cestodes are comparable to measuring tapes, these worms are called tapeworms. Majority of the parasitic cestodes to humans are endoparasites, meaning we can find them inside our bodies, particularly our intestines. So expect that their ova or proglottids will be seen in our stool sample. Most parasitic cestodes to humans are hermaphrodites, meaning one worm contains both the male and the female reproductive organs. Worm A belongs to the phylum Platyhelminthes, which includes the cestodes and trematodes, while worm C is a nematode or a roundworm. Take a look at the cross-section of the nematode. You have there a cavity that is called a xylem, while you don't have the same structure in the cross-section of the worm that is classified under phylum Platyhelminthes. So therefore, cestodes are acoelomic, meaning they don't have body cavities, unlike with nematodes. The same is true with trematodes. Trematodes are also acoelomic. Again, cestodes are acoelomic organisms, meaning they don't have body cavities similar to trematodes. They also do not have organs that function for digestion. That's why in the second bullet you have their alimentary tract is absent. So how do these worms survive? How do they get nutrients? Remember, they are found in our intestine. And in the lumen of our intestine, there are a lot of nutrients. These nutrients will just simply diffuse into the bodies of the cestodes. Their bodies are covered with cuticle or tegument, which will facilitate now the absorption of nutrients from the luminal contents of the intestines of the host. This cuticle or tegument will also protect the cestodes from the digestive enzymes present in the intestine. You know very well, pancreatic enzymes are drained into the duodenum to help in the digestion of food. And these enzymes can actually cause harm to these worms. And what will protect these worms is the cuticle or tegument covering of their bodies. The bodies of the cestodes have to be dorso-ventrally flattened so that it would be easy for the nutrients in the intestine to diffuse into their bodies. Just like in the case of trematodes, they have the protonephridia for their excretory system, and they have this nerve ganglion present in their scolex, and from the nerve ganglion, you have there the emergence of two lateral nerve cords. This is the head or the scolex of the cestode. As you can see, you have here a ring of nerve ganglion, and arising from the nerve ganglion are two lateral nerve cords. The bodies of cestodes are segmented, and these are the parts of their bodies. The scolex, that's the equivalent to the head of the parasite. The scolex is followed by the neck, and the neck is followed by that long structure which we will call as the strobila. And please do appreciate the strobila have segments, and these segments are referred to as proglottids. The general parts of the body of the cestodes include the head or the scolex. This head and scolex are provided with suckers and rostellum for attachment. I will be elaborating these structures on the succeeding slides. Then the head is followed by the neck. The neck contains actively dividing cells, and it is the neck that will give rise to the proglottids of the strobila. Let's start with the scolex. The scolex is provided with suckers. One of them is pointed by the red arrow. Suckers are also known as acetabula. And let's count the number of suckers present in the scolex of cestodes. We have 4. The other one is not shown in the picture. Aside from the suckers and acetabula, the scolex of some, I have to emphasize some, cestodes are provided with a protusible structure which we call as the rostellum. If the rostellum is present, it may or may not be provided with hooklets. So the point is, not all of the parasitic cestodes to humans have rostellum. If they do, some of the rostellum are armed, meaning they have hooklets, and some of them are unarmed. The rostellum and the suckers are used by the cestodes for attachment into the walls of the intestine. Take a look at these two scolices. You have there the presence of 4 suckers or acetabula. The scolex on the left side has a rostellum, and its rostellum is provided with hooklets, while the scolex on the right side of the picture is devoid of rostellum. After the scolex, we have the neck. The neck of the cestodes contains actively dividing stem cells, and the main function of the neck is to give rise to the proglottids of the strobila. The point is, if you are a doctor and you are managing a case of cestode infection, it's very important to have the stool exam repeated after the completeness of the therapy. And the medical technologists in the laboratory should be able to see the presence of the neck and the scolex in the stool sample of the patient. And that would indicate effectiveness of the therapy. Why? Because if the medical technologists will only see the proglottids and the scolex and the neck are still inside the intestine of the patient, the neck can still give rise to the series of proglottids in the strobila. So take note, to establish completeness and effectiveness of therapy, the scolex should be isolated in the stool sample of the patient. The third portion of the body of the cestodes is called the strobila, and the strobila is composed of series of proglottids. And there are 3 types of proglottids. The one that is closest to the neck is the immature proglottid or immature segment. In the middle of the worm, you have there the mature segment, and at the distal portion of the worm, you have there the gravid proglottid or gravid segment. The immature proglottids are named such because these proglottids do not have yet the mature male and female reproductive organs of the cestode. So it basically looks empty. And these proglottids are found near the neck or scolex of the cestode. The immature proglottids are followed by the mature proglottids. These proglottids would contain the mature male and female reproductive organs of the cestode. Cestodes are hermaphrodites. It means that they both have the male and the female reproductive system organs. Let's start with the male reproductive organs. The red circles are their testes, and these are the ones that produce the sperm cells. The sperm cells are drained into the vas efferens Effreens. This is another vas efferens. The vas efferens will all fuse together to form the larger vas deferens. The terminal portion of vas deferens is dilated and it's called the serous sac. This is one of the testes. It's difficult to appreciate the vas efferens because they are very small. So, we'll identify the next structure. This is the vas deferens. The dilated portion of the vas deferens is the serous sac. And the serous sac will drain into the genital pore. Testis, vas efferens, vas deferens, serous sac, and the lateral genital pore. Let's now discuss the organs that belong to the female reproductive system of cestodes. Draining the sperm cells from the genital pore is the vagina. And the vagina will drain the sperm cells into the circular structure which we call as the ootype. I hope you can remember from the trematode chapter that this ootype is the site where the ovary, the vitellaria, and the Melis glands would drain their products. The ootype is usually surrounded with Melis glands, and the products of the Melis glands will help in the formation of the shell of the ovum. What is also draining into the ootype is the paired ovaries and the vitellaria. The yellow-colored structure is the uterus. So this uterus is also connected to the ootype. So the ovary will release the egg cell into the ootype. The Melis glands will secrete products and as well as the vitellaria, and their products will form the shell of the egg. And the egg now will be transported to the yellow-colored uterus. This is the vas deferens. Below it is a thin tube, which is the vagina. The vagina will transport the sperm cell into the ootype, which is surrounded by the Melis glands. The ovaries will also drain into the ootype and as well as the vitellaria. And this ootype now will form the eggs of the parasite, and the eggs will be transported into the uterus. Vagina, ootype, which is surrounded by the green-colored Melis glands. Then, you have here the paired ovaries also draining into the ootype. Then, you have the blue-colored vitellaria. Also connected to the ootype is the uterus, which will now contain the eggs that were formed in the ootype. We have already discussed the immature and mature proglottids. Gravid proglottids are easy to identify because they are basically composed of prominent uteri filled with eggs. A is the Scolex. It has 4 suckers or acetabula. It is also provided with an armed Rostellum. Letter B is the neck. Letters C, D, and E are parts of the strobila. Letter C are immature proglottids. Letter D are mature proglottids. You can already appreciate in there the testis, the uterus, the vitellaria, and the ovary. Then letter E are gravid proglottids. You have there the prominent uterus that is filled with eggs. Proglottids arise from the neck. Do you know that as the cestode will form the gravid proglottids, these gravid proglottids will detach from their bodies so that they can release their eggs. So that will result to the shortening of the cestode. But that is not a problem to them because the neck can always give rise to new proglottids. So the mature proglottids will eventually become the new gravid proglottids. That's why it's very important to isolate the scolex and the neck to ensure effectiveness of therapy against cestode infection. There are two groups of cestodes. We have the Pseudophyllideans and the Cyclophyllideans. Both have bodies that are dorso-ventrally flattened and are segmented. We will try to compare Pseudophyllideans and Cyclophyllideans based on their scolex, their proglottids, and ova. Let's start with Pseudophyllideans. The scolex of Pseudophyllideans are spatulate in shape. So they basically look like that of the spatula that we are using in the laboratory. And their scolices are not provided with suckers or acetabula and Rostellum. So what will now function for their attachment to the intestinal wall? The slit-like grooves that are called as bothria. The ova of Pseudophyllideans are operculated and they have this posterior knob. I know some of you are already asking, how can we differentiate the ova of Pseudophyllideans from the ova of Clonorchis and Opistorchis? We will discuss that later on. Take note, ova of Pseudophyllideans are operculated and they are provided with posterior knob. It's also important to take note that upon discharge from the body of the human host, the eggs or ova of Pseudophyllideans are unembryonated. They have to stay first in the water before the larva will emerge from the egg through the operculum. The scolices of Cyclophyllideans are globular or spherical in shape, and they are provided with acetabula or suckers. Rostellum may or may not be present. The ova of Cyclophyllideans are non-operculated and they are already embryonated upon discharge from the human host. We have already differentiated Pseudophyllideans and Cyclophyllideans based on their scolices and their ova. Let's now try to differentiate Pseudophyllideans and Cyclophyllideans based on their mature proglottids. It's very important to take note that the Pseudophyllideans are provided with uterine pores. So please check for the uterine pores in the picture. Since they are provided with uterine pores, they can just release their eggs through these pores and there's no need for their gravid proglottids to detach from their bodies. Another important thing you need to take note of is that the genital pores of Pseudophyllideans are found in their midline or ventral side. So I want you to imagine a cestode, a Pseudophyllidean that is lying flat on a surface. The genital pore can be found on its midline ventral side. So you have to flip the worm so that you can see the genital pore. Please take note of the location of the genital pore in relation to the uterine pore. This is a mature proglottid of a Pseudophyllidean. You have there the midline or ventral genital pore, and you have there the presence of uterine pore. Uterine pore is absent in Cyclophyllideans. This is another picture of the mature segment of a Pseudophyllidean. This is its midline or ventral genital pore, and this is its uterine pore. I want you to identify what is this. Yes, that's the uterine pore. How about this one? You're correct. That's the genital pore. And since the uterine pore is present, and the genital pore is found in the midline or ventral side of the proglottid, then we are sure that this mature proglottid is from a Pseudophyllidean. Let's take a look at the life cycle of a Cyclophyllidean. This is the adult worm and it is found in the small intestine of the infected human host. Cyclophyllideans do not have uterine pores. So, the only way for them to release their eggs is to what? Allow their gravid proglottids to detach from their bodies. And the process is called apolysis. That's why what is usually seen in the stool sample of patients infected with cyclophyllideans is their gravid proglottid. But take note, in some cyclophyllideans, their gravid proglottids are usually fragile, so they might rupture while they are still inside the intestine, and what is seen in the stool sample is the ovum. But the thing is, since they do not have uterine pores, they must undergo the process of apolysis, the detachment of the gravid proglottid from the bodies of the cestodes. And since Pseudophyllideans are provided with uterine pores, apolysis is not seen in this group of cestodes. So the next question is, why do Pseudophyllideans still have their necks if they are not undergoing the process of apolysis? I want you to imagine one gravid proglottid of a Pseudophyllidean. That gravid proglottid will release the eggs via the uterine pores. So eventually, that gravid proglottid will become empty and it will disintegrate and it will detach from the body of the cestode or Pseudophyllidean. That is different from apolysis. Apolysis is the detachment of a gravid proglottid containing the eggs from the body of the worm. In the case of Pseudophyllidean, it is only when the gravid proglottid will degenerate after releasing the eggs that the gravid proglottid will detach from the body of the worm. So that's why the neck is still important for Pseudophyllideans. Pseudophyllideans release their eggs via their uterine pores, while cyclophyllideans have to discharge their gravid proglottids in the process called apolysis because they do not contain uterine pores. This is the life cycle of Diphyllobothrium latum. This is one of the two parasitic cestodes to humans that are classified as Pseudophyllideans. This is the adult worm. It is found in the intestine of the infected human host. And since the worm is provided with uterine pore, so they can release the eggs via their uterine pores, and what will be seen in the stool samples of the patients infected with Diphyllobothrium latum is the ovum. Take note of the name, Diphyllobothrium, dibothrium, because this is a Pseudophyllidean and it is provided with two bothria. Another thing that I want you to take note is that the genital pores of Cyclophyllideans are found on their lateral borders, while the genital pores of Pseudophyllideans are found in their midline or ventral side. So take a look at the structure labeled as genital pore. It is located on the lateral border of the mature proglottid. So therefore, this mature proglottid belongs to a Cyclophyllidean. Let's take a look at this mature proglottid. It has a uterine pore and it has a midline genital pore. So therefore, these mature segments belong to a Pseudophyllidean. Let's check this proglottid. It has no uterine pore and its genital pore is found on its lateral border. So therefore, this proglottid belongs to a Cyclophyllidean. Now, let's try to differentiate Pseudophyllideans and Cyclophyllideans based on their gravid proglottids. Gravid proglottids are known to contain prominent uteri. The uteri of Pseudophyllideans are usually convoluted in appearance, just like what is shown in the two photos, and they are said to be in rosette formation. While the uteri of Cyclophyllideans have lateral branches, just like what is shown in the two photos. Let's check these gravid proglottids. They have uteri in rosette formation or convoluted uteri. So therefore, these gravid proglottids belong to a Pseudophyllidean. Let's check this gravid proglottid. It has a uterus with lateral branches. So therefore, it belongs to a Cyclophyllidean. Please also check for the laterally located genital pore. So this will serve as a summary of the differences between Pseudophyllideans and Cyclophyllideans. So I want you to pause this portion of the video and start to review whether you have understood my discussions or not. Let's try to discuss the life cycle of one of the two Pseudophyllideans that can infect humans. This is the Diphyllobothrium latum. The adult stage of the parasite is found in the small intestine of the infected human host. And since the human host harbors the adult stage, then the human host is acting as the definitive host. Diphyllobothrium latum is a Pseudophyllidean, and Pseudophyllideans are provided with uterine pores, so this parasite can release its ova via its uterine pores. So that's why what will be seen in the stool sample of the patient infected with Diphyllobothrium latum is the ovum. This ovum is unembryonated upon discharge. It has to stay first in fresh water before a ciliated stage will emerge from it. And this ciliated stage is called the coracidium singular, coracidia plural. Now, if this coracidia will be ingested by small crustaceans such as the Copepods, the coracidia will become the procercoid larva. So that makes the small crustaceans as the first intermediate host. Small fishes usually feed on Copepods. So if these infected Copepods will be ingested or eaten by fishes, the procercoid larva will become the more slender and longer plerocercoid larva. So the small fishes in the life cycle of Diphyllobothrium latum are acting as the second intermediate host. These small fishes will be eaten by larger predatory fishes. In the body of the larger predatory fishes, the plerocercoid larva will just emerge out from the body of the small fishes, and this plerocercoid larva will insist themselves in the organs of the larger predatory fishes. And since there is no progression to another stage in the bodies of the larger predatory fishes, then these fishes are just acting as paratenic hosts. Eating raw freshwater fishes will now predispose us to develop the infection with Diphyllobothrium latum. What is the diagnostic stage of Diphyllobothrium latum? It's the unembryonated ovum. Please recall that the ova of Pseudophyllideans are operculated and they have this posterior knob. The infective stage of Diphyllobothrium latum is the plerocercoid larva. How many intermediate hosts are there? 2. Copepods or small crustaceans and small fishes. I'll give you 10 seconds to answer the following questions. Timer starts now. Humans are the definitive host of Diphyllobothrium latum, while we have two intermediate hosts in the life cycle, the Copepods, which will harbor the procercoid larva. Second, freshwater fishes, which will harbor the plerocercoid larva. Plerocercoid larva can also be referred to as sparganum. Diagnostic stage, unembryonated ovum. The infective stage is the sparganum or the plerocercoid larva. And the large predatory fishes are acting as parathenic host. These are the first intermediate hosts of Diphyllobothrium latum. We have the Copepods, especially those under the genus Cyclops and Diaptomus. And these are the second intermediate hosts of Diphyllobothrium latum. We have the Pike, the Burbot, the Perch, the Arctic char, and our favorite, salmon. So eating these fishes raw will predispose us to develop the infection. We have here in this picture the adult stage. Beside it is the ovum. It is operculated and it has a posterior knob. The ciliated stage is the coracidium. Then beside it is the procercoid larva that we can find in copepods or small crustaceans. And the longer plerocercoid larva which we can find in the organs of freshwater fishes. Diphyllobothrium latum ova, operculated, and it has posterior knob. And the operculum has just opened to release the ciliated stage, which is the coracidium. This is a plerocercoid larva encysted in one of the organs of a freshwater fish. Diphyllobothrium latum is also known as the fish tapeworm because we basically acquire it through eating raw fish meat. It's also referred to as the broad tapeworm because its proglottids are broader than they are taller. The height is around 2 to 4 millimeters and the width is around 10 to 12 millimeters. And the adult worm is easily identified because they have that dark central marking in their proglottids. And that dark central marking is actually the uterus. The adult worm is usually yellowish in color and it can be as long as 3 to 10 meters. That's why this worm is considered to be the longest tapeworm that can infect humans. With that length, they can have 300 to 400 proglottids. And their gravid proglottids are easily identified because of the dark central markings. And they can stay in our body for 5 to 10 years. At this point, let's just have a review. Diphyllobothrium latum is a pseudophyllidean. Whatever characteristics of pseudophyllideans we have discussed in the introduction will all be applicable to Diphyllobothrium latum. So let's start with the Scolex. The Scolex is spatulate in shape and it has no suckers or acetabula and Rostellum. Instead, it has slit-like grooves which we will call the Bothria. This is the picture of the Scolex of Diphyllobothrium latum. Please do appreciate the presence of Bothria on both sides. Two Bothria, Diphyllobothrium. The ova of Diphyllobothrium latum or Pseudophyllideans are operculated and they have this small posterior knob. I intentionally put side by side the photos of the ovum of Diphyllobothrium latum and the ovum of that of Clonorchis and Opistorchis. As you can see, both are operculated and both have posterior knobs. So how will you now differentiate one from the other? Please recall that we described the ova of Clonorchis and Opistorchis to have that thickened portion at the part in the egg where the operculum is fitted. And you don't have that thickening at the portion where the operculum is located in the egg of the Diphyllobothrium latum. You can also rely on micrometry to differentiate the ova of Diphyllobothrium latum and that of Clonorchis or Opistorchis, because the ova of Diphyllobothrium latum are way bigger than that of Opistorchis and Clonorchis. This is the mature segment or proglottid of Diphyllobothrium latum. This is one of its testes. The testis would drain into the vas efferens. The vas efferens will fuse together to form the larger vas deferens. And eventually that vas deferens will dilate to form the serous sac. And the serous sac is continuous with the midline genital pore. So I have just discussed the male reproductive system organs of Diphyllobothrium latum. Now, we will talk about the female reproductive system of Diphyllobothrium latum. This is the genital pore. This is where the serous sac will drain the sperm cells. From the genital pore, the sperm cells will be transported through the vagina. And the vagina will drain into the ootype. That ootype is surrounded by Melis glands. Aside from the Melis glands, what will also drain into the ootype are the paired ovaries. And this arrow is showing us the point of drainage of the ovary into the ootype. And then, take a look at this structure pointed by the red arrow. That structure connects the ootype to the uterus. And you know very well that the uterus of Pseudophyllideans are provided with uterine pores. In the mature segments or proglottids of Pseudophyllideans, in this case the Diphyllobothrium latum, the genital pore is located in the midline or ventral side of the worm, and you have there the presence of the uterine pore. Midline genital pore, vagina. The vagina drains into the ootype that is surrounded with Melis glands. This is one of the lobes of the ovary. This is the uterus. Please appreciate its connection to the ootype because the ootype is there to form the eggs, but the eggs are stored into the uterus. Then, the uterus of Diphyllobothrium latum is provided with uterine pore. Located on the same area where the testes are the Vitellaria. And it's very difficult to differentiate the Vitellaria from the testes. I cannot find a good picture of the mature proglottid of Diphyllobothrium latum in the internet, so I will use the mature proglottid of Spirometra mansoni, another pseudophyllidean that can infect humans. We have here the following structures. This is the midline genital pore. This is the uterine pore. This is the uterus. And as you can see, the uterus is spiral in shape. That's the reason why the organism is named Spirometra mansoni. If this is the mature proglottid of Diphyllobothrium latum, the uterus should be in rosette formation. This is one of the lobes of the ovary. And this is the ootype. Pointed by the red arrow is either the Vitellaria or the testis of the parasite. We will now describe the gravid proglottids of Diphyllobothrium latum. Again, what is prominent in the gravid proglottid is the uterus, and as described a while ago, Pseudophyllideans have convoluted uteri, and their uteri are in rosette formation. So take a look at the picture of this gravid proglottid. You have in the center the convoluted uteri. Again, The uteri of the gravid proglottids of Diphyllobothrium latum are convoluted and they assume the rosette formation. Patients with Diphyllobothriasis may not show any signs and symptoms of the disease. If they will become symptomatic, they will just be complaining of abdominal discomfort, loss of weight, vomiting, diarrhea, and malnutrition, which are non-specific to Diphyllobothriasis. Remember, this worm can be as long as 10 meters. And while inside the intestine, they might curl their bodies and cause obstruction. The chance for obstruction increases as the number of worms will also increase. Another important thing you need to take note is that Diphyllobothrium latum competes with the host for vitamin B12. Vitamin B12 is absorbed at the ileal segment of the small intestine, and that is also the area where we can find the adult stages of Diphyllobothrium latum. The deficiency of vitamin B12 will now lead to what we call as megaloblastic anemia. This is a picture of the adult Diphyllobothrium latum. Please appreciate the dark central markings which are representing the uteri in the gravid proglottids of the worm. Just try to appreciate how long the worm could be. So if this worm will curl its body in the lumen of the intestine, it can really cause obstruction. Diphyllobothrium latum can compete with the human host for vitamin B12. And vitamin B12 is important in the formation of red blood cells. So if you lack vitamin B12 because you have Diphyllobothriasis, then most likely that will result to a form of anemia. Vitamin B12 is also important for the maintenance or formation of myelin sheath. So in the case of Diphyllobothriasis, there's a probability that there is a problem in the formation and maintenance of the myelin sheath of neurons. There was this episode in House MD, I think it was in season 3, and the title of the episode was Insensitive. The girl in the photo had an accident, but the rescuers were surprised to find out that even if metals have already pierced through the thigh of this woman, she did not feel any pain. The doctors who were working in the hospital where House MD was working had a hard time figuring out what was causing this patient to lose her ability to feel pain. And at the last part of the episode, House MD tried to open up the abdomen of the patient. He did not even ask the anesthesiologist to administer anesthesia because the patient will not even feel the pain of the surgery. And House MD was able to recover the worm that is shown in the photo. Take a look at the worm. It's dorso-ventrally flattened. And later in the episode, the worm was identified as Diphyllobothrium latum. And it's the main reason why this girl has lost her ability to feel pain because of the problem in the maintenance of the myelin sheaths in her neurons. I would just like to correct something on that particular episode of House MD. Take a look at the Scolex of the tapeworm that was removed from the intestine of the patient. It has a globular-shaped Scolex, which is not characteristic of a Pseudophyllidean. The Scolex of Pseudophyllidean is usually spatulate in shape and it has bothria. Diphyllobothriasis can cause megaloblastic anemia. Megaloblastic anemia is characterized by the presence of MAC macrocytes in the blood, large red blood cells. Red blood cells with mean corpuscular volume of more than 100 femtoliters. Why do you think red blood cells will become macrocytes in cases of megaloblastic anemia? Shown in the picture is the maturation series of red blood cells. They start as pronormoblasts, then eventually this pronormoblast will become the normal mature red blood cell. What do you notice? The younger stages of RBCs are bigger than the mature stage because these immature stages are still active in undergoing mitosis, and every time they undergo mitosis, they reduce in size. Thus, the mature stages are smaller than the immature ones. Vitamin B12 is important for DNA synthesis. And you know very well that before a cell can engage itself in mitosis, it has to duplicate its DNA. In the absence of vitamin B12, DNA synthesis is not possible. So what will happen? The immature large red blood cells will not be able to undergo mitosis. Thus, they will mature with a large size. There is that another form of anemia that is also characterized by deficiency of vitamin B12, and that is pernicious anemia. Pernicious anemia is characterized by the production of autoantibodies that are directed against the parietal cells. So what are these parietal cells of the stomach? They are the cells responsible for the production of hydrochloric acid and intrinsic factor. Now, what's the importance of the intrinsic factor? Take a look at the red arrow. It's pointing to the ingested vitamin B12. This red arrow is pointing to the parietal cell of the stomach, and this parietal cell is producing the blue-colored intrinsic factor. The produced intrinsic factor will bind to vitamin B12, and it will accompany vitamin B12 as the vitamin B12 courses through the gastrointestinal tract until such time that they will reach the ileum. The intestinal cells of the ileum have specific receptors to intrinsic factor. So if the intrinsic factor will bind to these receptors, the receptors now will internalize the intrinsic factor and even the vitamin B12. So intrinsic factor enhances or facilitates the absorption of vitamin B12 in the ileum. In patients with pernicious anemia, they are producing antibodies against their own parietal cells. So the parietal cells will be destroyed by their antibodies, so expect that they cannot anymore produce intrinsic factor, and that will have an effect on vitamin B12 absorption, thus leading to vitamin B12 deficiency, and their red blood cells will also become macrocytes. So how will you now differentiate a case of megaloblastic anemia from that of pernicious anemia? You check the gastric fluid of the patient. If the gastric fluid is acidic, that will give us an idea that the parietal cells are intact and you're dealing with the case of megaloblastic anemia, probably secondary to diphyllobothriasis. But if the gastric fluid is alkaline, then that should raise the suspicion that the patient could be a case of pernicious anemia. The recommended drug for the treatment of diphyllobothriasis is praziquantel. And just like what I've told you, the Scolex must be recovered to ensure full recovery. In the event that the Scolex is not recovered, a repeat stool exam should be done 3 months after the treatment. And if that stool exam will turn out negative, then that will somehow give the physician the idea that the treatment is effective. How to prevent diphyllobothriasis? All freshwater fishes should be thoroughly cooked. if you really need to, eat them raw, might as well subject them to freezing for 24 to 48 hours at a temperature of negative 18 degree Celsius because that can kill the plerocercoid larvae. Another important prevention step is to properly dispose human waste. We will be talking about the second pseudophyllidean that can cause disease among humans, and that is the Spirometra mansoni. The definitive host of this parasite includes dogs, cats, and other mammals. The adult stage of the parasite is seen in their small intestine. What is discharged in their stool is the unembryonated ovum. Because the parasite is a pseudophyllidean, it has uterine pores. This unembryonated ovum will embryonate in the water and will release the ciliated stage, which is the coracidium. This coracidium will be ingested by copepods. Inside the bodies of the copepods, the coracidium will become the procercoid larva. If the copepods are accidentally ingested by fishes, reptiles, and amphibians every time that they will drink water, the procercoid larva will transform to form the plerocercoid larva. So if the reptiles, amphibians, or small fishes will be preyed on by the dog and the cat and other mammals, that's the time they will develop the infection because they will acquire the infective stage, which is the plerocercoid larva. Humans acquire the infection with Spirometra mansoni in two ways. Number one, drinking water contaminated with infected copepods. What we are getting in this manner of or way of transmission is the procercoid larva. Once ingested, the procercoid larva will transform to form the plerocercoid larva, and that plerocercoid larva will migrate in our tissues. I want you to analyze that in this situation, the human host harbors the larval stage, and there is the progression from the procercoid to the plerocercoid larva. If the host harbors the larval stage or the asexual stage of the parasite, the host is acting as the intermediate host. So in the case of Spirometra mansoni, humans are intermediate hosts. The second way on how we can acquire the infection with Spirometra is eating raw second intermediate hosts, the fish, the reptiles, and the amphibians. And in this way of transmission, we are actually acquiring the plerocercoid larva. And do you know what will happen? Once we have ingested the plerocercoid larva will automatically penetrate the wall of the intestine and migrate to the different tissues in the body. I want you to analyze. The plerocercoid larva in this manner of transmission did not transform or progress to another stage. So, in that manner of transmission, we are acting as paratenic hosts because the larva did not progress to another stage. But if we acquired the infection through drinking water with copepods that have procercoid larva, we're acting as the intermediate host. The point is, what is causing us to develop the disease is not the adult stage. It's the plerocercoid larva or the sparganum. It's the migrating spargana, plural, sparganum, singular, that is causing now the clinical manifestations of sparganosis. These spargana may be located anywhere in the body, including subcutaneous tissue, muscles, breast, orbit, urinary tract, pleural cavity, abdominal viscera, and even the central nervous system. The clinical manifestations of sparganosis would greatly depend on what organ is affected by the migrating sparganum or plerocercoid larva. The migration of the sparganum in subcutaneous tissue is usually painless. But the thing is, if this sparganum will reach the CNS, the brain and the spinal cord, the patient may start showing neurological signs and symptoms. If the sparganum will reach the ear, that can trigger an attack of vertigo, and sometimes it can also cause deafness. They can also affect the eye and cause ocular sparganosis. This is a picture of the migrating sparganum in one of the membranes of the pleural cavity. This is another picture of ocular sparganosis. As you can see, the area where the sparganum was taken appears to be erythematous, swollen, and there is also that characteristic tenderness or pain. This is a picture of subcutaneous sparganosis. This presented as a nodule in the subcutaneous tissue of the skin. When the nodule was incised, the surgeon was able to find a sparganum or plerocercoid larva. Again, the plerocercoid larva can be found in different tissues in the body. They can be found in the brain and cause cerebral sparganosis. They can also be found in the subcutaneous tissue. In the picture, the organism was isolated from the subcutaneous tissue of the breast. We can use praziquantel to treat cases of sparganosis, but the recommended management is the surgical removal of the sparganum or plerocercoid larva. Now, how to prevent sparganosis? We have to boil the water that we are supposed to drink so that we can kill the plerocercoid larva present in the Copepods in the water. We have to cook thoroughly the meat of known second intermediate hosts to kill the plerocercoid larvae. We have to avoid applying poultice on inflamed areas of the skin. Health education and proper human waste disposal.